Okay, welcome to chapter 15. This first segment will take you through an introduction to the chromosomal basis of inheritance. Now, what we want to kind of start out is remember the two laws which were developed by Mendel. So remember, the two laws can be related at the chromosomal level as well as at the just genotypic level and alleles. So if we look at the chromosomes and we follow those through, we can kind of relate those laws as well. So remember, you've got these yellow round seeds, and remember their genotype is the following, right? So they are homozygous dominant for both of the traits. And then the green wrinkled seeds are homozygous recessive for both of the traits. So these are opposite opposing phenotypes and of course genotypes. So of course we know what the result is, but let's look at the chromosomes here. So if we track the chromosomes, you could pretty much say that 50% of the chromosomes in this individual are going to have all of the chromosomes in this individual are going to have the the yellow uh, allele, and all of them are going to also have the round allele. And then for this one, all of the chromosomes will have the allele for green color and the allele for wrinkled shape. Now, remember meiosis. Okay, so meiosis it's a process of cell division, which happens twice, which results in gametes, which have half as many chromosomes as the parents, right? So we have the gametes here. And then let's go ahead and go to generation. So the next generation, we can see that all the F1s, after they're produced, right? So after the mating of the gametes of the parents, it produces heterozygous individuals. So remember, both the traits are heterozygous. You technically should not have to do a Punnett square for this. But let's look at the process of meiosis during cell division in these heterozygotes. Okay, so we can relate this law of segregation and the law of independent assortment. So the law of segregation basically states that the two alleles for each of the genes that separate, they're going to separate during gamete formation, and you can see them separated down here. And then, of course, the law of independent assortment uh, basically states that the alleles of the genes on non-homologous chromosomes, you can see right here, they assort independently during gamete formation. And this is kind of related to at metaphase, you can have different lining up at the equation or at the equator, and then they of course separate independently because they're independent of one another. They're on separate chromosomes and they're far enough away from each other if they were on the same chromosome that they would not be transmitted together. So here we can see the results of metaphase 1 and then of course metaphase 2 all the way to the end of meiosis 2. Okay, so that's just a quick reminder and relating it to meiosis and the chromosome at the chromosomal level. So here we can see the results, right? So the F2 generation, remember the F2 generation is formed by crossing an F1 with another F1. And remember, they're both heterozygous for both of the traits. So the law of segregation really shows us that basically after fertilization, then recombining of all the alleles, it's random, okay, and then they're randomly separated during gamete formation. And then the law of independent assortment, we can see here, fertilization is going to ultimately result in the 9:3 to 3:1 phenotypic ratio as long as the alleles are not basically transmitted together, or the term is linked, which we're gonna see and describe in a later segment for this particular chapter. Now, let's talk about Morgan. Morgan was another scientist who did study subgenetics. And we're going to talk about one of experiments, but let's kind of do a little bit of background. Okay, so it's Thomas Hunt Morgan. He was an embryologist. So he actually came up with the first significant evidence or solid evidence that relates a specific gene with a specific chromosome. Now, Morgan's experiments were with fruit flies. Okay, so he used fruit flies. And after his experiments, there was evidence, right, strong evidence that chromosomes are on a location. Basically, those are the heritable factors. So there, these locations or genes really are on the chromosomes, and these are what are inherited from one generation to the next. Now, several characteristics make using fruit flies pretty, you know, convenient in the lab, right? So they produce many offspring. They can generate, or they can breed every 2 weeks. Okay, so they can generate a new generation relatively quickly. And they only have 4 pairs of chromosomes, so it's easy to track all the genes within the chromosomes. Now let's look at one of. Now, it's important to note before we discuss the experiments is that he actually identified the presence of what's called a wild type and a mutant phenotype. So the wild type is considered the normal phenotype, and the mutant, of course, is the one that's not normal. Now, in this case, in terms of the eye color, the normal phenotype is the red, and the mutant phenotype is the white. So, Morgan actually correlated the behavior of the alleles of a gene with the behavior of the chromosomes when they paired up. So, in his experiment, which we'll describe in the next slide, what Morgan did was he mated male flies that had white eyes. So remember, white eyes are the mutant, and the female he made it with female flies that had red eyes. And again, the red eyes are the wild type. Now, what he determined from this experiment was that the white-eyed mutant allele must be located on the X chromosome. And we'll see the results and how that relates to the chromosome pair and the behavior of both the gene, the allele, and the chromosome pair. Now, what he found also from this set of experiments is that it supported the chromosomal theory of inheritance. And this kind of dates back and relates back to Mendel's work with inheritance. So let's take a look at the so taking a look at this, we have the parental generation, which I just described. Males that, or females that have red eyes were mated with males that had white eyes. Remember, white eyes is the mutant. And what was the result was that all the F1 generation had red eyes. So maybe at that point, Morgan might have thought, well, maybe red eyes. Of course, it's the normal, right? So it's the normal phenotype. Maybe it's dominant as well. If it was a gene or these alleles were part of a gene that were located on an autosome. So then, of course, proceeding through to see the transmission and how it occurs, you want to mate 2 of the F1 generation. So taking 2 of these F1 right here and mating them, right? So this would make a mating between a male that had red eyes and a female that had red eyes. Now, what the F2 generation, what the results were the following. So all the females had red eyes, and about half of the males had white eyes, and half of them had red eyes. So looking at this a little bit further, if we look at the chromosomes, so these are how they're denoted. So we have wild type, which is just italicized W with a plus, and then the mutant is just the italicized W. So this kind of shows that relationship. So here we have the females that have red eyes, assuming that this female has both of the chromosomes, and he determined that this gene must be located on the sex chromosomes, or really the X chromosome, because it was an effect seen in males. Remember, males only have one X chromosome. So here we have the female with two X chromosomes with the wild type, and then the male, of course, he has the white, so he's got the mutant on his X chromosome. This is actually what his conclusion was. This is what he determined from these results up here. Is that here we can see the F1. Okay, so remember all the F1 are going to be red, but here, because females have, again, one of the X chromosome is going to go to the female, even though it's mutant, the female has the wild type, and that's what's going to be expressed. So the females have red eyes, however, they're carriers, they're heterozygotes. And then, of course, the males, if they have red eyes, then that one X chromosome must have the wild type, and because it obtained that from the female, right? So remember, they get segregated during gamete formation, and then they come together during fertilization. So then if we look at these right here, the Punnett square of the F2 generation, so again, these are heterozygotes. And really, males are not really termed heterozygote when we're referring to sex chromosomes and genes on the sex chromosome, but we'll talk about that later. So we have the male that has the Y chromosome, and then the X chromosome has the wild type. And then the females here, we have wild type and then again, carrier. And these are the results right here. So we can see that there was a 3-to-1 ratio of red to white, and half of the males had red and half of the females, or half of the males have white. And then none of the females were affected with the white. However, it must be noted that females can have white eyes if both their X chromosomes have the mutant allele. Okay, so it's important to remember that is very possible. However, you're going to see more white eyes in males than you will in females. Okay, so let's do a little quick reminder, going back to the sex chromosomes. It's important to remember that, females have one X chromosome or two X chromosomes, and males have one X chromosome and one Y chromosome. So in humans, this is the same idea. There are two sex chromosomes. The X chromosome is a lot larger than the Y chromosome, and males have one Y chromosome. Okay, so these are not necessarily homologous, although they are the sex chromosomes, and there's a small portion of the Y chromosome that could recombine with the X chromosome. However, there is really generally no recombining going on with these two chromosomes. Now, here's a couple karyotypes. So we can see this is a karyotype from a female that has two X chromosomes, and a male, one X and one Y is present. Now, again, it's important to remember that females have two X chromosomes, males have one X and one Y. Now, what this means is that each ovum or egg contains an X chromosome. And then it's important to remember that the sperm contain either an X or a Y. So remember, these are separated. Now, let's say this chromosome, 50% of the eggs are going to have this chromosome, and then 50% of the eggs will have the other chromosome. And then here, in terms of sperm, 50% of the sperm will have an X chromosome, and then the other 50% will have a Y chromosome. So it's really the male that determines the sex of the child. Now, other animals have different types of. So again, in humans, we have the XY system, which we just talked about. The insects have an X0 system. So the females have two X chromosomes, and the males just have one X chromosome, and there's no Y chromosome present there. So here you can see that there are 22 chromosomes in these particular insects. Now that might be different for different species of insects. Okay, so that's the X0 system. And then we have the XW system and some mammals. So here, this shows 76 chromosomes. However, the females have the ZW chromosome and the males have two Z chromosomes. So it looks like the males have the chromosomes that are more homologous than the females, right? And then here we have in other insects, we have the haplodiploid system. So the females are diploid and the males are haploid. Okay, so there's different systems for sex determination in different types of organisms. Now, in summary, we reminded ourselves of the law of segregation and the law of independent assortment that related to the segregation of chromosomes and, of course, the assortment of chromosomes within the cell. We talked a little bit about Morgan and his initial experiments where he identified the genes on the sex chromosome and in flies. And then we talked about the sex chromosomes and sex determination in various different types of organisms. Now, the next segment will be dealing with linked genes. Okay, so we're going to kind of expand on Morgan's work and then talk about linked genes and linkage maps.